Okay, so we are now on chapter 20, and we're talking about evolution. And really, the first concept we need to think about is getting back to our genetic variation, because we know that there are variants, and Darwin appreciated the fact that species don't conform to a given type, that there are a lot of variants due to these genetic variations. And there are many ways that evolution occurs. One way, which we're going to spend most of our time talking about, is natural selection. And natural selection depends on the fact that there are all of these different variants, and that these different variances are going to succeed in varying ways in terms of their surviving and their reproducing. And we're going to talk about it. So basically, this is all about you have all these different variance among individuals in a species. Some of them, due to their particular alleles, are going to do better in their environment and they'll survive more and reproduce more. And we're going to talk about that quite a bit. So we know that genetic variation creates new alleles, right? New versions of genes. And mutations, right, are what lead to these genetic variations. And we know that there's no rule saying if a mutation is good, bad, or neutral. They can be any of the above. We know they can happen in your body tissues. Somatic tissues is a fancy word for that. We know they also can happen in your reproductive cells or in your germline and therefore passed on to the next generation. And we know that during the process of meiosis, you have recombinations of your genetic material leading to new generations, new versions. And so all of this leads to genetic variation. And you can see here, right, let's say there's this allele, and then all of a sudden this mutation pops up. And let's say that another new mutation pops up. And then let's say that during meiosis, you then are going to have this mutant and this non-mutant recombining, and then that can create another new variation. So there are lots of different ways that these variants can arise over time, and they're important because they play a fundamental role in evolution. Now, some terms that we need to become familiar with, and we're going to look at these in more detail later on. But for now, I just want you to kind of be aware. A species, and we'll talk about this in more detail coming up, but these are basically individuals that are able to share genetic material through interbreeding. Right? So they're going to share alleles due to reproduction. We're actually going to go into this in much more depth in the next PowerPoint, but this is just sort of a beginning point. And then you want to think about, well, what does a population mean? Population is an interbreeding group of organisms of the same species living in the same geographical area. That geographical area is relative. You might say, for example, a population of students at a college, or a population of students in all of Massachusetts, right? Those are different populations, but they involve the same people possibly. And then a gene pool refers to all of the alleles that are present in all of the individuals in a species for a given population, right? So let's say we're looking at a population of students at UMass Lowell. If we took all of the alleles of all of those students, that would be the gene pool at UMass Lowell. And we'll look at this again in more detail, but these are terms you should be familiar with. Now, when we think about evolution, one thing we want to think about is allele frequency. And so to measure variation and how this variation might change over time, we're interested in what is the frequency of alleles in a population. And so here's my definition. Allele frequency is the number of a particular allele present in that population divided by the total number of alleles. So let's say that we have total number of alleles, and we're just interested in how many are of X subtype. You take X divided by the total, that gives you the frequency. This is an example we're going to look at in class, so take a look. We'll talk about it in class together. So again, allele frequency is the number of copies of a given allele divided by the total number of alleles in a population. And here's another question we're going to look at in class together. So now we want to think about evolution. And there are lots of different ways evolution can come about. And when we think about evolution, we're looking at this fact that the frequency of alleles, these traits, in a population can change from one generation to the next. And when they do change, this is what causes evolution. So basically, we're thinking about evolution as a change in the genetic characteristics of a population over time, and specifically the change in allele frequencies. Both of these are ways we think about evolution. And when we think about evolution and allele frequencies, we have to go back and think about this Hardy-Weinberg principle. So Hardy and Weinberg were two scientists working in the early 1900s, who developed this mathematical model to analyze the consequences of matings in the individuals of a population. And they were looking at the entire population. They wanted to know what would happen if everyone had the capacity to breed with each other, right? So all individuals, all possible genotypes. They imagined that breeding would occur randomly. So they're saying you take all of the gametes that are possibly being produced in this entire population, in this whole gene pool, and then they're randomly coming together, seeing what could happen. Based on that randomness, they're then calculating the frequency with which you would see certain genotypes in that population. Sort of like doing a Punnett square, but instead of for this man versus this female, it's for all of the males versus all of the females in the entire population. And when we do this, we know there are lots of situations in which there are more than two alleles for any given gene. But here we're going to look at the simple way to do this. Let's just assume for the sake of argument that there are only two alleles for any given gene. And so we're going to look at gene A, and we're going to say there are two alleles, A1 and A2. And if there are only two alleles in that population, then the percentage has to equal 100%. So the percent of the time you see A1 plus the percent of the time you see A2 has to equal 100% because the total is 100. And we know that the only options are A1 and A2. Therefore, all of it has to equal to 100%. We're going to say the frequency of A1, like any algebraic equation, we're going to give it a letter. We're going to call it P, and let's say A2 is Q. And then the frequency of each of those, again, equals 100%. And here we're just saying, instead of looking at percentages, we're just going to say the number 1. And then a decimal point would be anything less than 100%. We know that because individuals are diploid, there are 3 possible genotypes you could have. You could be homozygous A1, homozygous A2, or heterozygous A1A2. The frequency with which you see A1A1 would simply be P, the frequency of A1, times P, the frequency of A1, which would give us P squared. Same thing for A2, but now it's Q squared. And for A1A2, it's actually 2PQ. We know the frequency of A1 is P. We multiply that by the frequency of A2, which is Q. But as we'll see in the next slide, we don't know if the male is giving you P or the male is giving you Q. So there's 2 different ways you could get PQ, and because of that, we multiply it by the number 2. So it's 2PQ. And so this is what I meant by a Punnett square of everything in the population, because you're basically saying, well, there's all of these eggs in the population, and there's a certain frequency of those eggs that have, let's say, big A, and a certain frequency that have little a. So here they're saying big A, little a instead of A1, A2. And then here you have all the sperm in the population. There's a certain frequency that has P, a certain frequency here with Q. And so now you can see that if we have big A, we're multiplying P times P, which gives us P squared. Here you can see if you're little a, again, you're Q squared. And then there's two different ways we could get big A, which is why we have 2 times PQ. This graph on the right is showing you genotype frequency on the y-axis and then allele frequency on the x-axis. Now, this part of the figure, there is a little bit of an error with. I'm going to add it in right now. This is supposed to, this top line for allele frequency is for big A, which we know is represented by the variable P. And then this bottom row is going to be little a, which is represented by the variable Q. Now, what you can see here, and I'll keep my colors consistent with what's here. If you have, so we're looking at little a, if you have 100%, right, because this is little a, of the little a allele, then you're going to have 100% of this homozygous little a. Whereas on the other hand, if you have 100% of the allele frequency for big A, which is over here on this side, right? This is big A, then you're going to have 100% for big A. And obviously anywhere in between, it's just going to drop down as the allele frequency goes down this way, and the other one will do the same in the other direction. And then you can see that if you have 50% of each, that's when you are going to have, and I don't have this color here, so bear with me, as purple is pink. When you have 50% of each, that's when you're going to get 50% of PQ. And so for the Hardy-Weinberg principle, it's really, again, assuming that everything is random. And so again, for the population to conform to this equation, no evolution can be acting on the population and mating has to be random. So it's basically a way to tell you what would happen if everything is random. And when all of a sudden things aren't random, then his equation all of a sudden doesn't work. And that's how scientists can tell that there's some change in allele frequencies in a population. And so it's interesting to people when we find situations in which it doesn't work with this equation. And if it's not working, that tells us there are evolutionary forces at work. So Hardy-Weinberg principle is sort of your null hypothesis. It's saying everything's random. And when things do not conform to this principle, then we know evolution is acting on that gene, or there's some non-random mating that's occurring. So again, the Hardy-Weinberg equilibrium, this model only talks about when certain conditions are met. And these conditions basically mean no evolution is happening, right? So one condition is there's no differences in survival and reproductive success, right? i.e. there's no evolution. So when there's no evolution happening, Hardy-Weinberg theoretically could, right, hold true. It also is important for this principle to hold true that you have a very large population, because sometimes you have random changes occur more frequently in small populations. And so this really only holds true in very, very large populations. It also only holds true when nobody is coming or going from the population, because the minute you have immigration or emigration or any people moving, then you're going to see changes in the allele frequencies. It only works when there's no mutations that can then lead to new allele frequencies, new alleles, and evolution. And it only works when there's random mating. So basically, the Hardy-Weinberg principle holds true if all of these work. So we only see this kind of theory work when every condition is met. The minute that one of these doesn't work, that equation does not fit. And again, it's a way for scientists to determine, well, if it is fitting or it isn't fitting. This helps us to determine if evolution is happening. So there are many ways that evolution can occur. One of the primary ways evolution can happen is through natural selection. And so we're going to spend most of our time talking about natural selection. But again, there are many different ways that evolution can occur. So natural selection is the change in allele frequency over time, which is basically our definition of evolution. But in this case with natural selection, it's due to that allele's impact on survival and reproduction. And natural selection says that the individual who is best adapted to survive due to beneficial alleles that they then pass on to their offspring, right? And the next generation will then be enriched with the same beneficial alleles. So individuals survive because of these beneficial alleles and then pass them to the next generation. And that next generation then preferentially will have that beneficial allele. When we think about this, we need to think about the definition of biological fitness. This does not relate at all to how big and strong you are. It only relates to the ability of an individual to produce offspring. It's usually measured relative to other individuals in the population. So it's sort of like, how fit is this individual compared to that individual in the same population? You can compare numbers of offspring each individual produces and then say, oh, this one is more fit than that one. Another term we want to be familiar with is adaptation. An important point, these are heritable traits. So things that can be inherited based on your genetics. That increases that individual's fitness. Again, this refers to biological fitness in a particular environment, right? Because sometimes you're more fit in one environment versus another, depending on what the environment is and what the gene is. And again, relative to other individuals who lack that trait. So adaptations increase fitness, right? We know fitness relates to reproductive success and survival, because survival leads to reproductive success, but really it's just reproductive success. And the assumption is if you have reproduced, you had to have survived. We'll look at this in class together. So natural selection requires a few steps, right? Step 1, traits have to vary. There have to be different individuals that have different traits in the population. Step 2, some differences have to be heritable, meaning that these differences we're seeing have to be able to be passed on to their offspring. Number 3, in each generation, more offspring are produced than can survive. And only some will survive long enough to reproduce, and some will produce more offsprings than others. And finally, some variants will lead to increased fitness. And again, fitness relates to the ability to reproduce. And obviously, if you reproduced, you had to have survived. And evolution through natural selection is the logical outcome of those steps we talked about. So basically, all those steps we talked about, you can boil it down to this. Evolution by natural selection occurs when there are variations that can be inherited because they're based in genetics. And these differences, these variants, lead to differences in survival and reproduction. And what happens is that these traits, which are inherited and allow you to survive and reproduce more, then will increase over time in the population, leading to evolution, which we know is the change in the allele frequencies, and also the change in the distribution and or the mean character traits in a population over time. There are a lot of misconceptions out there when we talk about natural selection and evolution. I am listing some of them here because you might possibly in your head have this own misconception. I want to make sure we clear it up. If you have any questions, let me know. The first misconception is that, so these are actually what is true, right? So these are, people sometimes have misconceptions that these are not true. So the first one is individuals don't change, because there is this misconception that individuals do change, and it's not true. Individuals never change. When we talk about evolution, we're seeing a change in the allele frequencies, and therefore a population as a whole changes. When we think about evolution, don't think about individuals changing, think about the population changing. Individuals, when they're selected by natural selection, they're not changing. They're simply producing more offspring who survive in comparison to other individuals. Evolution is not goal-directed. It simply will favor certain individuals who happen to be adapted to that environment at that time due to the presence or absence of certain alleles. So it might differ in a given year or a given environment. And it's not necessarily producing more complex organisms. In fact, complex traits are routinely lost or simplified in time. And there's no such thing as higher or lower organisms. Every organism is well adapted to its environment, right? So for example, we're not necessarily any higher or lower than bacteria, because some bacteria can live in incredibly severe environments that we would not be able to live in, right? They're adapted to their environment, we're adapted to our environment. We'll look at this in class together. So we've talked about natural selection and that there are these variations that can be inherited, and that especially thinking about differences in survival and reproduction. And now we're going to look at one specific type of variation, and this variation will be in skin color. Here you see a small land animal, and you see there are differences in skin color. On the far left, we have very light skin color, moving all the way to the far right where we have dark skin color. And let's say that the original population, most of the individuals were sort of a mean skin color, somewhere in the middle in this lighter range. So again, on the y-axis is the frequency of individuals. So the more frequency here is right there at the mean, and then they taper off as you get to the light and the dark color. Now, what can happen is there are many different types, different patterns of natural selection. There are examples where we see stabilizing selection, where you're actually selecting against the extremes and you're selecting for this mean middle ground for the organism. And a lot of this you can think about, well, it depends on the background the animal lives on. So think about how an animal is spotted when it's eaten and attacked so that it can't survive. It's spotted when it stands out from its background. But if the background is similar to the organism that has that mean skin color, then the background selects for organisms that blend in. The darker ones over here are going to stick out. They would be eaten. The light ones over here would stick out and they would be eaten, but the middle ones do well. And so this is called stabilizing selection, where you're selecting against the extremes and you're selecting for that mid ground. Directional selection will shift you in one direction or the other. So you still have this general bell-shaped curve, but maybe you're shifting toward having a darker colored mouse if it's a dark background. Or hypothetically, on the flip side, what if you had a light colored background? Then it would shift toward this direction, right? It can go in either way. Disruptive selection selects against the mean. So in this case, let's say you had a light colored background and you had a dark colored background, but you didn't have anything in between. Then you would select for the lighter mice and the darker mice. They would each do well, and then the middle ground, they would not do well and they would not survive. This is called disruptive selection. And then you would end up with two populations, one on the light, one on the dark, that had their own bell-shaped curves. So again, different ways you can have natural selection, and we're going to look at a video in a moment that specifically relates to this example of animals and their fur color. Okay. And now we are going to watch a video, and I'm just going to play the portion that's relevant, which is the majority of it, just clipping off the first minute and the last minute that aren't that important. It's also uploaded to Blackboard if you want to watch it there. And this is a nice video that looks at the evolution of pocket mice, and it's going to be just like the example we just looked at with skin color. So here we go.
Across the American Southwest, golden deserts dotted by cacti and brush stretch for miles. Yet here in New Mexico's Valley of Fire, the landscape changes dramatically. Patches of black rock interrupt the sand, remnants of volcanic eruptions that occurred about 1,000 years ago. The eruption spewed a river of lava more than 40 miles long across the desert. As the molten rock cooled, it darkened, leaving any creature dependent upon camouflage in serious trouble. In the complex battle of life, one of the constant struggles is between seeing and not being seen, the evolutionary game of hide and seek. And we've come here to the Valley of Fire in New Mexico, a battlefield, to find one of the tiniest soldiers and what it can teach us about how evolution works. On the desert sands, the rock pocket mouse blends in perfectly, its light-colored fur concealing it from predators. But on dark lava, the same fur makes the mouse stand out, attracting the many creatures that see it as food.
These mice are the Snickers bar of the desert. They're eaten by foxes and coyotes and rattlesnakes, and certainly by owls, and maybe even occasionally hawks. And most of those predators are visual predators.
So what happened to the pocket mice that found themselves on this new terrain? When I accompanied biologist Michael Nachman onto the lava, it doesn't take long to find out.
Oh, this one's closed.
Nachman has been collecting mice unharmed in traps. And it's a dark one.
It is, yeah.
Now, are most of the ones you find up here dark?
Almost all of them.
Not only have the mice here evolved to be as dark as the rock,
Shake them into the bag.
the color change has occurred precisely where it will conceal them from hunters. Now, a bit of a white underbelly, too.
That's right. All of the dark ones here and on other lava flows have a white underbelly, and presumably there's no selection for dark on the belly because predators are coming from above.
Left to themselves, the mice show no preference for light or dark rocks. It's the predators that have made the difference.
The change in color over evolutionary time in the population is driven by predators weeding out the mice that don't match their background.
But how did the dark mice arise in the first place?
When a black mouse appears in a light population of mice, that is usually going to be due to a new mutation. And those are random and rare events.
To fully understand the pocket mouse transformation, Nachman moves from the lava to the lab. He and his team extract DNA from light and dark mice taken from one desert region. The aim? To find one or more genetic mutations that cause dark coloration. A mutation is a change in the chemical letters that make up our genes. It's a copying error that may occur when our cells divide. Mutation seems to mean that something bad has happened. Well, mutations are neither good or bad. Whether they are favored or whether they are rejected or whether they're just neutral depends upon the conditions an organism finds itself. So for the pocket mouse, a mutation that caused the mouse to turn black, that is good if you're living on black rock. It's bad if you're living out in the sandy desert.
The light mice are all on the bottom, here.
Fur color is a trait controlled by many genes. To figure out how dark mice evolved, Nachman focuses on how these genes differ in dark and light mice. One by one, the genes prove identical, but at last, something does turn up. The difference between dark and light mice boils down to a difference of 4 chemical letters in a gene called MC1R. Because the gene controls the amount of dark pigment in a mouse's hair follicles, a mouse with these mutations grows dark fur, which gives it an advantage on a dark background. But still, that's one mouse. How would its dark fur spread to a whole population?
This lava flow is about 1,000 years old. And so you might wonder, has there been enough time? It's only been 1,000 years. It's a very short period of time for a new mutation to come along and spread, and so that all of the mice on this lava flow are black, because really they all are.
Indeed, such a rapid spread of a mutation may seem unlikely until you do the math. And the reason is that while only one new mouse born in 100,000 may be black, hundreds of thousands of mice are born in any given year. And then those mice that are black have enough advantage that their babies do better, and they have more offspring, and their offspring have more offspring. And just about a 5% advantage, compounded year in and year out, can very quickly turn the whole population black, as we see today. If dark color gives mice a 1% competitive advantage, and you start with 1% of the population being dark, then about 1,000 years, 95% of the mice will be dark. If instead, the dark color gives them a 10% advantage, then it only takes 100 years. Thanks to Nachmann's mice, science has an example of evolution crystal clear in every detail.
What's exciting about this is that we have a system that's very simple ecologically. You have dark rocks and you have light rocks, and you have dark mice and light mice. It couldn't be simpler.
We know who the predators are, what the selective force is. We know precisely the genetic basis of what makes the mice have an advantage or a disadvantage depending upon where they live. All the pieces are finally together. It's a perfect illustration of Darwin's process of natural selection. In fact, it's more than that. For Nachmann's mice also counter a common misconception that evolution is a random process. Well, there is one random component, and that's the process of mutation. Mutations occur at random throughout our DNA. Every new organism is born with a new set of mutations. But while mutation is random, natural selection is not. Natural selection sorts out the winners and losers, and that's really what the whole process of evolution is driven by. But if natural selection is not random, would it produce the same result under the same conditions? It does. And here's proof. Rock pocket mice collected by Nachmann from other lava flows in other parts of the Southwest.
These are two different black mice, and they each evolved on different lava flows, and the lava flows are hundreds of miles apart, but the changes, the genetic changes that made these mice black, were different in each case. And what's amazing to me is how similar the black mice are. We didn't know when we started this whether we would find that there were the same genes or different genes, and we were really surprised to find that they were completely different genes, and yet if you look at the mice, they look almost identical.
Clearly, there are different genetic ways to make a mouse dark. But once the beneficial mutations appear, natural selection, the non-random part of evolution, can, under very similar conditions, favor very similar adaptations.
In effect, each of these lava flows is like rewinding the tape of life and allowing evolution to occur again and again. And in each case, we find the dark mice have evolved.
The rock pocket mice show us that evolution can and does repeat itself, and why evolutionary change is never ending. As environments transform, so must the species that inhabit them, adapting and readapting in the great and complex battle of life.
Now, another point to think about is that, while yes, natural selection is occurring naturally in the environment, there's also times where humans have artificially selected. And when we think about genetic engineering and how a lot of people are against genetic engineering of plants and animals, we want to also make note that we've actually been genetically engineering things for years by, instead of allowing organisms to breed naturally as they would, by forcing them to breed with the ones we want them to. And so in this example here, what they've done is they're doing research on corn, and corn is producing, there we go, pen. And these corn are producing oil, and they might select for corn that has very high or has very low oil content. And what they do is they just say if they're selecting for high oil content, then over time, over these generations of reproduction, as we move across the x-axis, we're just only allowing the high oil content plants to breed with each other until eventually over time you have this very, very high oil production. On the flip side, you can do the opposite with the low oil production. If you keep crossbreeding with organisms that are low oil over time, you can remove the oil content. So again, this is another means of selection, but this is done artificially by breeders purposefully choosing which organisms to breed with others. Another type of selection, which you're going to learn a lot more about in upper level courses, but we're just going to briefly talk about now, is sexual selection. And in sexual selection, we see animals that produce or maintain very metabolically expensive traits, right? So you can see here peacock with this beautiful feathers and wingspan, and here you see some animals that are fighting with each other with these horns, and these actually cost the animal a little bit, right? They're big structures. They don't necessarily have any role in the day-to-day function of this animal, and yet they're important. But what's interesting is that because these are so metabolically expensive, they also, in addition to the fact that they're metabolically expensive, they also can actually increase fighting and predation. Imagine if you have this beautiful wingspan, it's actually attracting predators towards you. And here you have antlers that are causing them to fight. And this would all decrease survival, right? It's metabolically expensive, and now you're fighting with other animals and you're possibly putting yourself out there as a target to be eaten. So why? Why would this happen? It doesn't make any sense when you think about natural selection, and it's everything about surviving and surviving in order to reproduce. So we know natural selection acts to increase survival, and this up here doesn't seem to do anything about survival. But now we're talking about sexual selection, and sexual selection acts to increase the probability of reproduction. And it turns out that females will choose mates based on ornament. Here you can see an example of ornament, right? These beautiful feathers. It actually indicates to the female, if they see these beautiful feathers in a male, that this is a healthy organism that's been able to produce these beautiful feathers. Or they choose a mate based on armament. And here you can see that you have these structures that lead to good fighting, and a female might choose the organism that wins the battle with their beautiful antlers. And this trait, right, it's a compromise between survival and reproduction, because we already know up here we saw, wow, this is decreasing survival, but now when it comes to sexual selection, it's actually increasing the probability of reproduction. And so this is an interesting dichotomy where it's a little bit of cost and benefit, but still we see these structures, so clearly the benefit of reproduction outweighs the cost relative to the survival. And so most of what we've talked about with evolution has been about natural selection. But there are many, many ways, other factors, things that can lead to evolution. And unfortunately in this course, we don't have a lot of time to go into these. We mostly focus on natural selection, but I'm going to briefly talk about these others right now. Another factor that can impact and lead to evolution is migration. Migration means that you have alleles in a person or an individual that's moving back and forth with them as they go from one population to the next. This movement of these alleles is referred to as gene flow, and this leads to homogenization of populations. They're mixing with each other. It makes populations more similar to one another over time, and it can reduce genetic differences. So this is another way that evolution can occur. Another way that we haven't talked about, although it's important, we know, is mutation. Mutation is a source of new alleles that other forces can act on, like natural selection or sexual selection. Without mutation, you would never have variation. So we know this is fundamental to allow for variation and therefore evolution to occur. And the last one we're going to talk about, and we'll do a little bit more for this, is this random process known as genetic drift. This is a random change in allele frequencies that occurs from generation to generation. Frequencies of certain alleles can go up, they can go down, they can stay the same, and all of this is happening by chance. It tends to be more pronounced in small populations because random things tend to happen in small populations. Think of when you toss a coin, heads and tails, 50-50. Theoretically, if you toss a coin 100 times, well, you should get 50 times head, 50 times tail. But if you only toss it 4 times, just by sheer randomness, you could end up getting 3 heads in a row and then 1 tail. Does that mean your coin is loaded? No, it just means you only had 4 chances to look. If you kept doing it over and over, you would have seen more likely that it hit the 50-50 mark. So you see these random events more frequently in small populations, but genetic drift can occur in all populations. So here's an example, genetic drift in small populations. In a small population, extreme departures from the expected outcome are very common. And this can lead to what we call fixation of an allele, meaning that you can actually go 100% toward another one. So let's say that you had 2 alleles in a population, and in this case, you're seeing that instead of being at this 50% allele frequency for, let's say, let's say we're looking at the allele frequency for big A versus little a, right? You start off somewhere in the middle, 50% big A, but over time, over these generations, in this small population, you could end up with 100% big A. Or over time, you could end up with 0% of big A. And this is just due to randomness. And then we say fixation. Fixation shows here because if you have 100%, if there's nothing of the other allele, that's what we call fixation. And these things tend to happen more in small populations, these random events. Whereas when you have a large population, shifts in allele frequency tend to be much smaller. So you don't see a lot of fixation taking place in these populations. And in fact, you do maybe a little bit of fixation at population size 40, but when you all of a sudden get to population size 400, you no longer see that fixation. So the bigger the population is, the less likely you are to see these dramatic shifts and departures from where you started. So here again, you start around 50% and you can get to fixation, but you can see in a population of 40 versus 4, what we saw before, it would take more generations before you would reach fixation. But now in a population of 400, even bigger, you don't see any of these reaching fixation. They tend to stay closer to what you started with. We'll look at this in class together. And so when we think about evolution over time, so this is a timeline, present toward past. You start with a population of organisms, and over time, let's say something separates these organisms. So now you have this new group A and this other group B. And in this example, they're being separated by this mountain range. Well, if enough time passes, Group A, moving on its journey, can accumulate mutations that are different from Group B, moving on its journey. And then you could lead to differences between Group A and B over time. And this is how speciation occurs, which we're going to talk more about in the next section. And when you think about molecular evolution, you also can think about individual molecules. And here, again, we're looking at time on the x-axis. And then on the y-axis, we're looking at the number of amino acid substitutions per 100 residues. And residues is a fancy word for amino acids. So it's basically out of 100. So how many of them are, how many of these changes are we finding? And some genes can accumulate more mutations over time than others. So you can see, for example, this is interesting, histone genes have not accumulated many mutations over time. And it could be that the original gene that was evolved many years ago was working very well, and it doesn't allow for new mutations. Maybe new mutations cause this gene not to function anymore, so you don't see a lot of change. Whereas hemoglobin, as we talked about, is an example where you have an expansion of gene families and you see lots of changes occurring over time. And so different proteins, different genes that code for these proteins can accommodate differences in the amount of change that can happen over time. All right, and that is it. So we started with the Hardy-Weinberg principle as our null hypothesis. This is sort of random. What would happen if everyone randomly got together and there was no sexual selection, there was no mutation, there's no evolution? What would happen, right? Then we talked a little bit about the principle of evolution and allele frequency changes over time. We talked about natural selection, adaptations, biological fitness. And we talked about the fact that the misconception that evolution is progressive is wrong. It is not progressive over time. Obviously, there are other concepts we talked about in here as well. Those would not be excluded on an exam. These are just some of the key highlights of what we talked about.